Hi, my name is Madeline Mirador. This is my mini exam video 4 for Genetics 311. So here we have the Watson and Crick DNA model. So as you can see, DNA takes on a double helix shape. There are going to be two phosphate backbones, as you can see here. So these are these strands right here creating the double helix. And then in between here, we have our base pairs. So within DNA, we have nucleotides. So we have the phosphate group here, and then the sugar, and the base. So on the sugar, there are carbons that attach to the base and the phosphate. So obviously the 1' carbon here attaches to the base. The 2' carbon actually attaches to a hydrogen in DNA, which distinguishes it from RNA because RNA attaches to an OH group. And then the 3' carbon actually attaches to the next 5' phosphate group and the next part of the DNA helix. And then 4' and 5' here obviously go to the next phosphate group. And so now these nucleotides can be put into two groups, purines and pyrimidines. Purines are known for having two rings, as you can see here with adenine and guanine. Pyrimidines have one ring, as you can see here with thymine and cytosine. These group together as base pairs. Adenine pairs with thymine and guanine pairs with cytosine. And as you can see here, I've represented hydrogen bonds with these lines here. So adenine and thymine have 2 hydrogen bonds, while guanine and cytosine have 3. So we come back to this DNA model because I wanted to showcase the base stacking here. So the bases will stack one on top of each other in a DNA molecule like this because they're so hydrophobic. And then also, DNA strands will run anti-parallel. So one strand will go 5' to 3', and then the other strand will go 3' to 5'. So they run in opposite directions of each other. This molecule also shows a right-handedness, which occurs most often in nature, but as you can see, it turns up and to the right here. And then lastly, there are these major and minor grooves in the model here. And these major is obviously bigger than minor, which means it just has more space to interact with the proteins in DNA, and it has more base pairs in each groove as compared to minor. So if we put DNA into a higher concentrated salt solution like this, we'll find that the salt will actually stabilize the DNA and increase the TM, the midpoint of thermal denaturation, because it will neutralize some of the negative charges on the phosphate backbone, and so that will stabilize the DNA. And then here are my references.